Okay, so we're going to continue on with active immunity. We're going to talk specifically about the B cells and T cells. So when we're thinking about active immunity, we're thinking about a learned response to an antigen. So it's part of the immune system that learns to recognize an invader, usually by parts of the invader in some way, shape, or form. And then given those antigens that it's presented with, it will remember that disease causing organism. Hopefully, there'll be multiple antigens per given disease. And the more we have, the better our defense against future attacks from that disease. So we've already been through this slide, the lymphocytes. We're looking at the B's and T's. So a number of things can happen from those lymphocytes, and these are all steps in the active immune response. We'll talk about the B's and the T's in a very simplified way. So our B cells, lots of different B cells. Like the T cells, they learn what's bad. In their case, they're going to build specific antibodies to the antigens that they learn about, and they'll store the blueprint for those antibodies in the library. Their job is to make those antibodies. So memory cells, they're going to be waiting in the library until they are needed, and then they will reproduce at a rapid rate. Memory, depending on what it is, some will last forever in your body, or as long as your body lasts. Others will fade over time. That memory won't hold. But the B cells are going to create antibodies. So what are as antibodies? So we can call antibodies immunoglobulins. They're interchangeable, those phrases. So the immunoglobulin is the term for the protein carbohydrate molecule that binds to a specific antigen. And again, for a disease-causing organism, there may be multiple antigens that these B cells produce antibodies for. So we have some schematics here, or cartoon drawings, so basic structure of the antibody. We think about the chains, and some are heavy and some are light. So we've got these heavy chains forming here. So you kind of have a Y structure, kind of the stem, and then these are the active sites here. So if we look at the antibodies, what's common on all of them is these sections here. So we've got some light and some heavy, but they're constant. The variable region up here is the important part. You're going to have two of those per antibody. And these variable regions up here, if you look at this, are what binds to the antigen. So part of the learning process is I need to find something in the antigen that I can make or have recognized by this section. So you're looking at something that will be sticky to an antigen. So we have an antigen binding site, an antigen binding site. So whatever that antigen is, the body will design a binding site that will attach to that antigen. And that's what takes the time, that's what takes the energy, that's what takes the effort. So B cells produce these antibodies, and their job is to stick to an antigen that they recognize. So if we look at these in more of a space-filling molecule, again, we've got that kind of Y structure in both of these. This is kind of, we've seen before, we've got the coils and sheets associated with the protein folding. So sheets here, lots of sheets here, lots of coils going through. So this is the common area, and then we have our variable areas here at the end. These are where the antigens bind to. This is more of a space-filling molecule with individual amino acids being represented by each little dot. And as you can see that these are huge molecules. We have the variable heavy, variable light, VLVH, variable light heavy. So this is our binding site. Binding site. These blues are the constant areas that you'll find on every antibody. It's the binding site that is designed for the specific invader. That's what takes time. So, as we said, B cells produce those antibodies. Their job is to bind to the antigens. We have a immunoglobulin here. We've got the blue is constant, the red is those antigen attacking areas. So we've got the antigens sticking on there. They're going to fit specifically to the antigen. Say this is the surface of a bacterial cell. It's got a certain protein that the antigen will recognize. That antibody attaches to. Now you can have multiple antigens for a specific disease-causing organism. So we've got 4 different types here. We've got a different antibody associated with each of those. So we've got 4 different types of B cells that will recognize this foreign body and attack it. The more different strains we have, the more antibody types, the more likely we are to put this disease-causing organism out of business. So once the things are stuck, those antibodies will attract the non-specific, the second line of defense, by the active, the third line of defense recognizing and say, hey, over here, get this one. We have the rest of the immune system attacking. So it helps kind of pull those things out from hiding in some way. They're stuck, they're going to be removed. Different parts of the body will remove the covered antigens and dispose of them, get rid of them for recycling and disposal. So if we're looking at our antibodies, the things that might be produced by the B cells, we have 5 different classes. We have the IgGs. Well, these are the ones we usually think about with colostrum. The big ones in circulation. In the mom cow, they'll be taken out of circulation, put into the colostrum, passed on to the babies for the next generation. IgA, major immunoglobulin in bodily secretions, protect the mucous membranes. So you have antibodies in there that are help slow down the entrance of those foreign invaders, epithelial surfaces, skin. IgEs bind the mast cells and basophiles as part of allergic reactions and parasitic infections. So we need to have those types there. IgMs associated with early immune responses. IgDs found in B lymphocytes and needed for full maturation as we move through. T cells. They have a slightly different approach, but they still kill. So they learn what's bad, just like the B cells, and then it's more of a personal attack. Instead of standing across the room and throwing Spider-Man webs at it like the B cells, the T cells have a more personal, more hands-on attack. So the T cells will learn what needs to be attacked. They'll wait in the library until they're called, just like the B cells. They're memory cells. They can last for years and years, and then they reproduce when they're needed. So as soon as they are called into action, they're going to split into 2, 4, 8, 16 cells, and that whole army is going to attack that antigen, that invader, that specific bacteria that needs to be removed. So cell-mediated immunity, you have the T cells, different types of T cells kind of working together. So you've got the cytotoxic T cells. They're going to find, grab, or destroy that invader and help create the memory. The helper T cells kind of tell the cytotoxic cells what to do. It's more of like hey, over here, you got to get this one. So moving through, and then the suppressors turn the other T cells off, basically put them back in their cages. If these T cells get too crazy, they're going to really rip apart the body. So this is a good diagram to kind of go through and kind of look at how things might work. We have the B cells kind of in here, and then you've got the T cells over here. So antibody mediated immunity, we have extracellular microbes, we have the B cells secreting the antibodies attack, um, and then these are um, eliminate the extracellular microbes. So identifying them, getting them kind of tied down and removed from causing problems. So with the cell mediated, you have the non-specific, we have them engulfing the uh, problem. The helper T cells are going to go over to that white blood cell and say, hey, you need to destroy what you've engulfed, and it'll work together with those to destroy. So activate the macrophages to kill the phagocytosed microbes. So basically turning each of these pockets into a lysosome. So all the acids and peroxides and enzymes go in there and destroy whatever in those membrane-bound vesicles, kind of like a lysozyme. Or what we're dealing with now with the coronavirus, we have a virus that goes into a healthy cell, takes over that cell, and begins to multiply within that cell. Our cytotoxic T cells are going to go and kill that infected cell. They're going to realize that, hey, that is a defect. We need to get rid of it. And this is where we get run into problem with this new infection. Cells are they kill the infected cells and eliminate the reservoir of infection. The viruses hiding inside a cell normally can't be found, can't be seen by the immune system. So by pulling those out, by getting them loose, we can eliminate the reserve of that infection. And that's what we have to do to resolve things. But as you can imagine, if you're killing the cells that have been taken over by the virus, you're killing what was formerly a healthy body cell. That's all for B's and T's. Any questions?